Coming to the next slide about the microbial classification of infectious diseases and the viruses, again they are acellular, obligate intracellular microorganisms. They are consist of a specific nucleic acid known as DNA or RNA, and some of them are surrounded by a coat called as a capsid. So, the viruses which are enveloped, they are covered by an capsid, and viruses which are not covered, they are called as a non-enveloped viruses. So, in the next slide we will see how the viruses have been classified now based on the DNA and RNA, and also based on their outer coat, whether they are enveloped viruses or non-enveloped viruses. So, if you see here the viruses, again just now I told you classified based on their nucleic acid material DNA and RNA, and also it has been classified based on the strandedness, whether they are single stranded or a double stranded, and also on the size and the shape of the capsid, whether it is been covered, enveloped or not covered or non-enveloped viruses. So, in the next slide again it is a continuation of the viruses classification. If you see in the next slide how it has been non-enveloped and enveloped viruses have been seen. Non-enveloped viruses are very dangerous, it is very difficult to treat, and they are very resistant. They are usually, like if you see the example, double stranded DNA, non-enveloped papilloma or adenoviruses, and double stranded RNA, it could be a Reoviridae, or single stranded RNA, it could be a calcivirus or a picornavirus. Single stranded DNA is again Parvoviridae. So, these are the non-enveloped viruses which are difficult to treat, and they are very refractory to the treatment. Coming to the enveloped viruses, it could be like if you see the figure, double stranded DNA, where we have a Poxaviridae vaccinia, herpes simplex virus and hepatitis B viruses. These are all enveloped viruses, and single stranded RNA viruses which are enveloped are again Paramyxoviridae, it could be measles, Orthomyxoviridae, it could be influenza, Rhabdoviridae, it could be rabies, and Retroviridae, this is very important again HIV, human immunodeficiency viruses, Arenoviridae and coronaviruses, Bunyaviridae and Togaviridae also are seen. Nowadays, hospital acquired settings, if you see the Flaviviridae viruses are common, again Paramyxoviridae is very Orthomyxoviridae is very common, where we have the H1N1 and other viruses. So, now this is the era where not only the bacterial infections, but also the viral infections has to be treated, and it has to be taken care very cautiously. If you ask me here, again the bacterial infections are getting treated with antibiotics, viral infections getting treated with antivirals, but the difference between these two main is that viral infections are very virulent and they can get spread very fast when compared to the bacterial infections. So, viruses can get spread very fast. So, we need to have a very good infection prevention and control practices in the hospitals to prevent this emerging and re-emerging viruses in the hospital. This is in short about the microbial classification of the viruses. Now, coming to the next slide about how it gets entry into the host cell with an envelope. This most of us knows, whenever the viruses or bacteria they come into contact with the host or a susceptible host, they get attached to the host and they secretes an enzyme, and this will, if you see in the slide, that cause absorption spikes of the virions, they get attached to the host cell and membrane fusion occurs, and the next step the nucleic capsid will be released into the cytoplasm, and viral envelope remains a part of plasma membrane, and this will nucleic acid separate from the capsid and it enters into the host chromosomal material. That is how it attaches, the viruses attaches and it transfers their nucleic acid material or the chromosomes and get integrated with the host chromosomes and cause mutations, and they come up with a new infections. So, next slide if you see here is about the fungi. Again I said fungi are not behind in the race, maybe we have more bacterial infections, multi-drug resistant infections followed by viruses. Now, even the fungi is in the race, where we have the candidiasis, and we also have hospital just because of not being taken care properly in the engineering control. We have come across the even aspergillosis infections in the ICUs and also the mucormycosis infections. So, this is again a very important entity where we have to differentiate between the candidiasis, the candida again whether it is albicans or a non-albicans group. Nowadays, most of the hospitals are having non-albicans infections, again I would rather say because of again the different variety of the treatment the patients are getting, the wide development in the field of medicine with the newer chemotherapy agents and newer immunosuppressants, where we are coming across now newer non-albicans candida, which is again becomes a very important in the hospital, and it has to be treated and addressed immediately. So, these fungi again have two forms, yeast and mold forms. Most of them are yeast forms which we see in the hospital. Fungi are dimorphic. This is particularly true for the pathogenic fungi, and mold grows as filamentous branching strands of connected cells known as hyphae. Molds usually we see in microbiology department, where once we inoculate onto the Subarotes dextrose agar on any fungal culture media, where the aspergillus or the candida they come up with a mold growth on the plates, where we can see the typical filamentous forms with the branching strands. This is the again the part of the fungi. After bacteria, the viruses and the fungi is also a very important thing, and we have to know at least the basics about the fungi which can cause hospital acquired infections like candida, candidiasis and aspergillus, mucormycosis. These are very common. At least these three things has to be known, and again the antifungals, the treatment for this is very important. Then fungi, once we have seen the fungal infections, now we see about the parasitic infections, though I would say it is not very important in the hospital setting, but again now these parasites at a community level has to be basic knowledge about the parasites have to be there. If you see in the next slide about the parasites, microbial classification of parasites, again they are single celled microscopic organisms again, and that can perform all functions of metabolism and reproduction. Some of the protozoas are free living, while others get parasitized for the nutrients and the life cycle. So, this is again the parasites. This is another class where it is very important. Coming to the next slide now, about the microbial classification of the parasites again. Helminths being a free living or a parasite, nematode, trematode and cystode, again this is important at the community level, I would rather say, and this has to be dealt again separately with the antiparasitic drugs. The next thing about the continuation of the microbiological classification of infectious disease is prions. Again it is an abnormal transmissible agent that are able to induce abnormal folding of normal cellular prions, proteins in the brain leading to brain damage and characteristic signs and symptoms of the disease. They are rapidly progressive and always fatal. Prions again it is infectious particle again which can cause degenerative brain diseases, and its role has been in the newer researches have been proved that it can cause many neurological problems. The continuation of this, if you see here the prions, how they can enter into the body, interact with the normal protein, and how the normal protein is converted to the abnormal protein, and how it causes the infections. So, at the end of this module, in conclusion, in this module 3 we have seen that. So, in conclusion we have seen in the module 3 about the microorganisms, their classifications, bacteria, viruses and fungi, and how it has been classified based on the morphology of their structures, gram positive and gram negative in gram bacteria, and viruses based on the nucleic acid material and the standardness and the replication type, and in the fungi again it is not behind, as I said, it is also coming up with very fast with the hospital acquired infections. So, this module was basically been we have been sensitized now with the outcomes of the patient, what will be the outcome if the immunity is compromised, and what would be the immunity outcome if the immunity is good, and the different types of microorganisms. Thank you.